to be here to share with you on some common issues we observed in evaluation of a complex API. Before I start, I would like to ask the audience, by the show of hands, who are currently involved in the development or manufacture of complex APIs. Here. Thank you. For this presentation, I will first give you some examples of APIs that are considered to be complex. I would then go over the challenges that are associated with these APIs, and also discuss some common issues that we observe. And then I will try to offer you possible solutions to address these issues. One thing I want to emphasize is that all of these issues, you can always link all of these to the critical quality aspect of the API. Here, I will show you some examples of the APIs that are considered to be complex. They can be peptides. I don't know, the font is so small. I hope you can see. So they can be peptides. Peptides have well-defined primary structure, which is in contrast to the next category, complex mixtures. For example, low molecular weight heparins, or they can be synthetic polypeptides such as glutamate acetate. They can also be polymers, for example, sevelamer carbonate. They can also be molecular complexes, for example, iron sucrose. In addition, they can be derived from a natural source, for example, conjugated estrogens. In general, the molecular structure and the impurity profiles for these APIs are more complex than small molecule drug substances. Therefore, usually they cannot be easily characterized by a typical analytical testing. In addition, their manufacturing process will have a great impact on the identity, purity, potency, and physical chemical properties. What is excluded in these presentations are biological drug substances. For generic application, they should be submitted through a biosimilar pathway. In addition, these presentations exclude low molecular weight, well-defined small molecule drug substances. I'm sure you are very familiar with the requirements for approval of a generic drug product. Previous speakers have touched on these. But for the purpose of my presentation, I'm going to, again, go through these. A generic drug product needs to be the same as ILD in the following areas: the same active ingredients, the same strength, dosage form, route of administration, condition of use, and the labeling with certain permissible differences. In addition, the generic drug product should be also bioequivalent to the ILD. So out of all of these requirements, the same active ingredients and the strength are directly linked to the APIs. Ultimately, the approval of a generic drug product should be based on adequate information to ensure the identity, the strength, the purity, and the quality of the drug product. For my presentation, I would like to discuss the areas of the drug substance that impact the drug product identity, strength, and purity. I will now move on to the challenges associated with these APIs. To illustrate the challenges, I will focus on three quality areas. The first is regarding to the identity. For drug substance that is a mixture, sometimes it is the identity of the drug substance may not be clearly defined. Again, for a drug substance that is a mixture, the strength of the drug substance or the strength of the API may be based on the whole mixture instead of basing on the individual component. Sometimes a bioassay, which is based on the mechanism of action, may be required. For drug substance, that is a mixture. Sometimes the line between active ingredients, concomitants, or impurities may become very blurry. Therefore, it is very challenging to differentiate active ingredients from concomitants or impurities. In the next few minutes, I will move on to discuss the common issues that we observe. I group these issues into two big buckets. The first one is related to the characterization regarding to the identity and the physical chemical properties, assay, and purity. The second bucket is related to their control strategies. For example, starting materials and raw materials control, or manufacturing process and controls. For in the areas of drug substance specification and their analytical procedures for sameness characterization or release and stability testing. And in general, if issues are identified for the drug substance specification and analytical procedures, these issues need to be resolved before drug substance retest periods can be established. I will now give you a little bit more detail for each of these elements regarding to their issues. The first one is related to identity and physical chemical properties. Usually, the issues are due to insufficient data or inadequate acceptance criteria. So one could address these issues by utilizing orthogonal analytical techniques to establish the identity of the drug substance. In addition, for drug substance that is a mixture, for example, enoxaparin or glatiramer acetate, it would be very beneficial for the applicant to identify structure signatures. In addition, the use of negative controls would be highly recommended. These negative controls are used to demonstrate the sensitivity and the selectivity of the analytical procedures used to used for drug substance release testing or sameness characterization. Usually, the negative controls are prepared by changing a critical process parameter that is outside the range for commercial manufacture. The negative controls are expected to meet the basic quality requirement, for example, molecular weight, but they are expected to fail at least some of the critical quality attributes. To justify the acceptance criteria for the identity, this is especially the case for drug substance that is a mixture. The applicant may be able to use data from analysis of multiple batches of RLD and use the data to justify the acceptance criteria. I will now move on to the issues related to assay. Usually, the issues are due to inadequate reference standard or its characterization. To address these issues, the applicant could address through a primary reference standard. Again, for drug substance, that is a mixture, and if there are no available USP reference standard, the applicant may isolate the API from RLD. And usually, the primary reference standard need to be fully characterized using a full battery of tests. Usually, the test is beyond the typical release testing. In addition, information should be provided. If you isolate the API from RLD, you should provide information how you isolated the API used for primary reference standard. And also, you should provide information how you determine the potency of the API. For example, for peptides, you may use a mass balance approach. In such case, you should provide the equation that you use to determine the potency. I will now move on to the issues related to purity. Usually, the issues are due to inadequate acceptance criteria or lack of adequate assessment of potential impurities. You can address these issues by adopting the requirements in the USP monograph if the monograph is available. Or you can refer to available FDA guidances or ICH guidelines. Or you can analyze multiple batches of RLD and based on the data to justify acceptance criteria for the impurities. And again, you could also based on the PharmTax study to justify the level of green impurities. To address the issues regarding potential impurities, one could acquire process knowledge through development studies or scale-up runs. In addition, data should be provided to support that the proposed analytical procedure is adequate, for method specificity to detect impurities. It's also encouraged to use orthogonal analytical techniques to assess potential impurities. I will now move on to the second bucket related to a control strategy. The first one is related to starting material and raw materials control. Usually the issues are due to an inadequate designation of starting materials or specification. So to address these issues, it's highly recommended that you refer to ICH Q11 or the clarifications provided in the Q11 Q&As. Recently, a training slide deck for the selection and justification of starting materials was published in ICH website. The case study, the slide deck includes 2 case studies, which I think are very useful for illustrating how to apply the general principles provided in ICH Q11 for selection and justification of starting materials. So if you have not checked it out, I encourage you to do so. To justify the starting material specification, I also want to draw your attention to the identity test. Usually, retention time by a single chromatography is not considered to be specific. However, if you use a combination of, say, HPLC retention time and specific optical rotation, that may be acceptable to be considered as a specific ID test. For justification of impurity controls in the starting material, data should be provided to demonstrate that the acceptable drug substance quality can be obtained using the proposed acceptance criteria for the impurities in the starting material. I will now move on to the issues related to manufacturing process and control. Usually the issues are due to insufficient details. This is actually very common. Or inconsistent information in sections S2.2, 2.4, or master batch record. Or due to inadequate in-process control or intermediate specifications. Or because that the critical process parameters are not adequately identified. Or because that the ranges for the critical process parameters are not adequately justified. So to address these issues, the applicant should provide clear, accurate, and consistent information with sufficient details in this application. Because of the complexity of the drug substance, usually there's a huge amount of information in the submission. However, that's not to be used as an excuse for unclear or inconsistent, inaccurate information. Also, I encourage that the applicant to conduct a development study to identify critical quality attributes and critical process parameters. The ranges for the CPPs should be justified. Frequently for drug substance that is a mixture such as enoxaparin or glycerin acetate, it is highly recommended that the justification of the CPPs be based on sameness data. Once the CPPs and the ranges of these CPPs are identified, the information should be provided in section S2.4. I will now move on to the issues related to drug substance specification. Usually the issues are due to inadequate acceptance criteria or missing critical tests. To address this issue, I encourage you to acquire process knowledge to identify critical quality attributes. And once you identify these critical quality attributes, they should be included in the drug substance specification. To justify the acceptance criteria in the specification, one can rely on the requirement for USP monograph if the monograph is available, or one can refer to available FDA guidances or ICH guidelines, or based on analysis of ILD data. In the next few minutes, I will show you some real examples. These are the actual deficiencies that we communicated to the applicant. I have provided quite a lot of examples in the slide, and I know I won't have enough time to go over all of them, but just see how many examples I can go through before my time runs out. Also, I want to emphasize for these deficiencies, I highlight the core issues in red, and the recommendation I offer are highlighted in blue. Another point I want to emphasize is that the recommendation by no means is the only way to address these issues, but nonetheless, we try to offer you like an approach, a way that you can address these issues. So the first example is related to lack of negative controls for drug substance as a mixture. For example, enoxaparin or glatiramer acetate. This is a very lengthy, busy slide, so I'll give you a minute to read. So essentially, what we are trying to recommend the applicant to do is use the negative controls to demonstrate the sensitivity and the selectivity of their analytical procedures for sameness characterization. These negative controls should demonstrate that they should cover that the critical process parameters that can potentially affect the structure signatures. The second example is related to impurity control for peptide drug substance. Peptides are known to have diastereomers as potential impurities, and these diastereomers are formed because of the manufacturing process or due to the presence of enantiomers in the amino acid starting materials. So to address the issue of these potential diastereomers, the applicant could provide data to support their analytical procedures for the related substances can detect these potential diastereomers. The third example is related to inadequate acceptance criteria for drug substance specification. Again, because of, I'm going to run through this quickly. In this particular case, the applicant proposed a limit of not more than 0.2% for the 3 specified impurities based on HPLC retention time comparison of between the generic drug substance and the RLD drug substance. However, just because the HPLC retention times are the same as the RLD, it doesn't mean that these impurities are indeed the same as the RLD. Therefore, the recommendation is that the applicant provide structural characterization data for these impurities in both of the generic drug substance and the RLD drug substance. And then, the information can be used to justify the acceptance criteria. So my time is running out, so I'm going to skip the rest of the examples. If you are interested, you can read them on your own. To recap, I have shown you some examples of the drug substances that are considered to be complex. I also discussed the challenges associated with these complex APIs, and I show you some examples of deficiencies that are common to these APIs, and I also offer possible solutions to address these issues. The issues that I presented today are associated with the identity, the potency, and the purity of the API. Ultimately, the identity, the strength, the purity, and the quality of the drug product need to be assured to support safety and efficacy. These also form the basis of a CMC recommendation. That's it. Thank you.